I'm going to talk to you about the work that we have done at the level of single cells, looking at mutations in splicing factors. First, and perhaps you are more aware in recent years that there has been a lot of studies showing how, as we age, we tend to accumulate many mutations, which points to, like the talk that we hear before, how this accumulation of new mutations in the cells makes that the genomes of the cells are different, and therefore some of these cells tend to expand into creating certain clones that upon the years then they could lead to disease. More interestingly, in recent years, we also seen that in the hematopoietic system, especially hematopoietic stem cells tend to also accumulate some of driver mutations that expand clonally, and then with the pass of the years, they lead to several hematological malignancies. Some of these mutations that are clonally expanding tend to appear in particular genes or particular gene groups. And one of the most interesting gene groups that I'm studying is splicing factors. As many studies in the past using TCGA and other cancer atlases, they have described how many times in different cancers, splicing factors acquire mutations that then lead to these aberrant splicing patterns that are driving the disease and also all the phenotypes that we observe in patients. Particularly in blood, we have seen, and blood is perhaps one of the most clear examples where many of the patients, in particular subtypes of the certain hematological diseases, they can have a high frequency of splicing factor mutations. Like in this case, we see splicing factor mutations in SF3B1 being present in more than 60% of the patients. SF3B1 got on interest because it's a core splicing factor which collaborates with the U2F subunit in order to splice and recognize properly the 3' splice sites. And the aberrant form of SF3B1 tends to have an aberrant recognition of the 3' splice sites, leading to several alterations, including part of the intron into the transcripts, which often leads to the creation of aberrant proteins and degradation by NMD. Therefore, we wanted to know how can we understand the relationship between the splicing aberrations and the cellular phenotypes that are observed in the patients with hematological diseases. And this has been very difficult in the past because there are not very good models that recapitulate hematological diseases in mouse. But more often, most of the studies that exist today with human samples rely on bulk data, which doesn't allow for the distinction of mutant and wild-type cells because we have just a mixture of these mutant and wild-type cells. But also, it doesn't let us to look at these complex phenotypes, these different cell types that we can observe in blood. Moreover, perhaps more interesting for this crowd, this studies in bulk also, even when we go to single cell, we still rely very much on the transcriptome done with short-read sequencing, which doesn't help us to reconstruct properly the isoforms that we can observe in the patients. With this thinking in mind, we developed GoT-Splice, which is based on a technology that our lab previously developed, which is called GoT, or by genotype of the transcriptomes. GoT basically builds on the 10x genomics platform in order to amplify a particular mutation of interest. But in this case, we, on top of this genotyping, we also added the long-read sequencing. And in order to have a good description of the different cell types, we included site-seq. So now we can measure from the same cell which proteins are in the surface of the cell to identify the different cell types. We can measure which are the identities of the different cells, are they wild type or mutant to the splicing factor mutation, and whether we can properly reconstruct the transcriptome using the long-read sequencing. So we applied this to a cohort of MDS, myelodysplastic syndromes, MDS, or also to a preclinical stage that is not considered malignant. I will tell you a little bit more about this in a bit. As a result, we were able to recover the expected cell types that we always observe in hematological diseases. But more interestingly, we also observed that there is a clear accumulation of the mutant cells, mostly in the erythroid progenitor cells. These erythroid progenitor cells tend to have, for the analysis that we did directly on the level of expression, defects on the translational machinery and also in the cell cycle. And we are able to integrate in the proteo-genomics data to identify how certain markers of expression, particular for erythroid progenitors, they start increasing with the pseudo time in the differentiation trajectory, but moreover, also the mutated cells tend to increase with the increase of the trajectory. But now looking at this phenotype of alternative splicing, well, we wanted to see how can we assess this using the long read. And normally with the 10x, you will probably have around just a fraction of the transcript when you want to look at it. But with the long read, you can now see the full picture, compare different isoforms across different cells, compare different isoforms across the same cell. And this is just a snapshot of the quality of data. In this case, particularly for SF3B1, we are able to reconstruct with very much clear. It's very clear how the 3' splice site is altered only in the long-read data, which is not possible with the short read. Moreover, when we compare the mutant versus the wild-type cells, we also see this bias towards an increase of cryptic 3' splice site usage only in the mutant cells. This is just a snapshot of what we see in this high quality of data. Now separating the cells of mutant versus wild-type, the change is very clear in the mutant cells. And now, looking to the differentiation trajectory, we are able to see clearly how certain isoforms are only particularly differentially spliced in some cell types versus others. But for instance, here we can see the erythroid progenitors or the material erythroid progenitors also expressing a block of differential spliced isoforms. And now, given that there are conditions in which there are clonal expansions apparent in healthy individuals before the disease, we also wanted to look at what is the effect on these patients that are completely normal, but that they have this clonal growth. And this is important because this has been observed that one of the most frequently mutated clones in these normal patients tend to also be splicing factors like SF3B1 or others. And moreover, these splicing factors also carry a risk to what we see in the development of the disease. So we applied GoT-Splice now to healthy samples with these clonal expansions of SF3B1. And what we observed was that even though it's not dramatically as in the case of MDS, we are able to see an accumulation of the mutant cells in these erythroid progenitors. Moreover, the effects of the splicing changes are also reflected in this pre-malignant condition where we see an increase on cryptic junctions, which many of them are also shared with the total number of junctions that are cryptically spliced in the patients that already have the malignant disease. And one interesting example of this is a gene known as BAX, which is particularly involved in the apoptosis of the cells. And in particular, BACS has this duality of different isoforms create have different functions, contrasting functions. So we describe BACS omega as being one of the key isoforms that gets misregulated already in the pre-malignant condition and leads to an anti-apoptotic function, leading to perhaps to explain why we see an accumulation in all the erythroid progenitors just dying. This was also tested experimentally with help of our collaborators. And in conclusion, what I wanted you to take home is that GoT-Splice is a very powerful tool that gives you an idea of the cell surface, the genotyping, but also of the full-length transcriptome in the single cells. And that there are certain mechanisms that are already present in the clonal, in the pre-malignant condition that then are reflecting as well in the splicing phenotypes when the hematological disease is there. Thank you.